I want you to picture something. You are standing in the Trans-Himalayan region of Leh Ladakh, India, and you're breathing
Yeah,
heavily.
you're absolutely gasping for air.
Right. Every single step you take feels like the last mile of a marathon because you are standing at an elevation that ranges anywhere from 2,300 to over 5,700 meters above sea level. It's what's known as an extreme cold desert ecosystem.
Which means hypobaric hypoxia.
Exactly, hypobaric hypoxia. That is the scientific way of saying the atmospheric pressure is so low that your lungs just have to work overtime to pull in a normal breath of oxygen. Plus, the winter temperatures there regularly plummet well below minus 20 degrees Celsius.
That's brutal.
It really is. You look around and the landscape is mostly barren. There's, very limited vegetation. Water resources are incredibly scarce. And because the atmosphere is so thin, the sun is just beating down on you with this intense ultraviolet radiation.
Basically, it's an environment that actively, aggressively discourages life. I mean, it really does.
Yeah,
And yet, life always finds a way to endure.
it does.
Surviving in that kind of harsh, unforgiving landscape requires some truly remarkable biological adaptations. Because of these extreme conditions, the lack of oxygen, the freezing cold, the poor food, only a very select few livestock breeds have actually managed to naturally evolve and thrive up there.
Which brings us to the true stars of today's deep dive. Today, we're unpacking some groundbreaking new research that looks at the DNA of the native cattle living in this extreme environment.
Yeah, the Ladakhi cattle.
Right. Our mission today is to, look under the hood of these animals. We're going to uncover the genetic secrets of the Ladakhi cattle, explore exactly how their bodies manage to survive, and figure out what makes the milk they produce so biologically unique.
And critically, understand why conserving their specific DNA is just absolutely essential for the future of global agriculture.
Yeah, that conservation piece is huge.
It is. I think it's worth establishing right away that this isn't just a story about, an obscure breed of cow in a remote part of the world. This is a masterclass in evolutionary biology.
A masterclass. I like that.
Because these Ladakhi cattle are one of India's unique indigenous populations.
Mhm.
And the key concept there is indigenous. I mean, they haven't been shaped by intensive artificial human breeding programs aimed at, like, maximizing profit or milk yield.
Right, nobody was tweaking them for dairy production.
Exactly. They've been shaped entirely by natural environmental selection over centuries. Nature was the only breeder, and survival was literally the only metric of success.
And based on the descriptions and the research, they are visually striking animals, too. Like, they aren't those massive, lumbering beasts you might picture when you think of a commercial dairy farm.
No, not at all.
They're quite short stature. They stand only about 75 to 80 centimeters tall. They have a very compact, cylindrical body, a small hump on their back, and this long, glossy hair to protect them from the biting cold.
Yeah, they usually come in black, brown, or black with white patches.
But the detail in this study that genuinely stopped me in my tracks was their efficiency. I mean, despite the hypoxia, despite freezing in the snow and surviving on this incredibly poor quality scarce forage, these animals still produce about 2 to 5 kilograms of milk every single day.
Which is an absolute lifeline for the people who live there.
Oh, for sure.
I mean, for the local communities in Leh Ladakh, land resources are incredibly meager. Traditional agriculture is exceptionally difficult due to the short growing season. So that daily milk production from the cattle is indispensable for their livelihood.
And their basic food security, right?
Exactly. It's a highly efficient biological system that basically converts sparse, tough scrub brush into vital human nutrition.
Now, I have to admit, my first thought when I read that number, 2 to 5 kilograms of milk, I was slightly skeptical, and maybe you listening are wondering the exact same thing.
It sounds low compared to commercial dairy.
Right. I thought, well, if food security is such a challenge in this region, why not just truck in a commercial dairy cow? Bring in a Holstein that can pump out 30 kilograms of milk a day. Wouldn't that solve the nutritional deficit immediately?
And that is the logical, purely economic question that policymakers actually ask all the time.
I'm sure they do.
However, biology really doesn't care about economics. The thing is, if you dropped a high-yielding commercial cow, like a Holstein, into Leh Ladakh, it wouldn't produce 30 kilograms of milk. It would likely perish very quickly.
Just from the altitude.
Mostly, yeah. A commercial breed simply does not have the internal genetic hardware to survive hypobaric hypoxia. Their lungs, their metabolic systems, they're designed for sea-level oxygen.
Oh, wow.
Yeah, so you add in the extreme cold stress and the poor quality forage, and their bodies would just shut down.
So they're basically like high-performance sports cars that only run on premium fuel on a paved track, whereas the Ladakhi cattle are off-road vehicles built for the rocks.
That's a really fair comparison. Indigenous breeds possess completely unique genetic resources. They have climate adaptation, disease resistance, incredibly efficient feed utilization, and reproductive fitness built right into their cellular structure.
Right.
And preserving these naturally evolved traits is becoming a massive priority globally. Because as the global climate changes, bringing more unpredictable and extreme weather, the resilience found in breeds like the Ladakhi cattle might be exactly what future livestock breeding programs need.
Like a genetic vault they can draw from to ensure sustainable agriculture.
Exactly. We aren't just saving a specific type of cow, we are preserving a biological safety net for our food systems.
Okay, so to understand exactly how they manage this incredible survival act, we really have to zoom way in. The researchers in this study didn't just observe the cows in a field, they looked at their DNA.
Right down to the molecular level.
Specifically, they focused on what they call candidate genes. Now, as someone who, doesn't spend their weekends in a genetics lab, I'm assuming a candidate gene isn't just a random snippet of DNA.
Far from random. Candidate genes are specific, pre-identified genes that scientists already know have a significant influence on economically important traits.
Okay, so they know what they're looking for.
Exactly. In the context of dairy cattle, researchers have spent decades mapping which genes are responsible for milk yield, protein content, fat percentage, the milk's cheese-making ability, and just the overall growth and health of the animal.
Right.
By isolating and looking at these specific candidate genes, scientists can identify genetic polymorphisms within the population.
Okay, let me see if I can translate genetic polymorphism into something more tangible for us.
Go for it.
Imagine the cow's genome is a massive biological instruction manual for building and operating the animal. Polymorphisms are essentially just slightly different versions or different spellings of the exact same instructions among individuals of the same species.
That is a really useful way to picture it, yeah.
Oh,
Though
yeah.
biologically, it is a bit more dynamic.
How so?
Well, those small spelling changes in the DNA sequence actually alter how the resulting protein folds and functions in the animal's body.
Oh, so a spelling change changes the physical shape of the protein.
Right, and that altered protein function is the mechanism that ultimately dictates the milk quality, or how efficiently the animal processes energy in the cold, or its overall immune response.
Got it.
Identifying these different genetic spellings helps us understand the physical differences we see in the herd and provides a foundation for scientifically informed breeding strategies.
Makes sense. And the methodology the researchers used to hunt down these spelling differences seems highly deliberate. I mean, they didn't just walk onto a single farm and draw blood from a bunch of sister cows.
No, that would ruin the data.
Right. The study notes they collected blood samples from 72 unrelated, true-to-type Ladakhi cattle scattered across 11 different villages in the region.
Yeah.
Why go through the logistical nightmare of traveling to 11 different high-altitude villages just to ensure the cows are unrelated?
Well, it's a fundamental requirement for a rigorous genetic study. Selecting unrelated animals from a geographically widespread area minimizes genetic bias.
Because otherwise you're just studying one family.
Precisely. If researchers only tested one or two herds in a single valley, they might just be documenting the genetic quirks or, inbreeding of one specific family line.
That wouldn't be helpful.
Right. By spreading the sampling across 11 villages and ensuring the animals are unrelated, they gather a truly representative picture of the entire Ladakhi population's baseline genetic diversity.
Okay, so once they secured those 72 blood samples and got them back down to the lab, they used a molecular genetics technique called PCR-RFLP.
Try saying that 3 times fast.
I know, it sounds like a random string of alphabet soup. Walk us through what is actually happening in the Petri dish here.
It sounds intimidating, but it's a standard and highly effective technique. Let's break it into 2 parts. The first part is PCR, which stands for polymerase chain reaction. You can think of PCR as a highly targeted molecular photocopier. A single strand of DNA is just too small to analyze effectively. So the researchers use PCR to target the specific candidate gene they want to look at, and they force it to replicate millions of times.
Until they have a sample large enough to actually study.
Exactly.
Okay, so we've photocopied the specific page of the instruction manual we want to read. What is the RFLP part?
So RFLP stands for Restriction Fragment Length Polymorphism. This is where we actually look for those different spellings you mentioned earlier.
Okay, how did they do that?
Researchers introduced restriction enzymes into the amplified DNA sample. These enzymes act as highly specific molecular scissors.
Oh, molecular scissors.
Yeah, they're programmed to only cut the DNA if they recognize a very particular sequence of letters.
So if a cow has genetic variant A, the scissors recognize that specific spelling and cut the DNA string into two distinct pieces.
Right.
But if the cow has variant B, the spelling is slightly different, the scissors don't recognize the sequence, and the DNA string remains whole.
Precisely. The researchers then run the sample through an electrical gel that separates the DNA by size.
So they can literally see the cuts.
Yes. If they see two small fragments, they know the cow has variant A. If they see one large fragment, they know the cow has variant B. It's a visually definitive way to know exactly which genetic variant that individual animal is carrying.
That is wild.
And importantly, they didn't just do this for one gene. Milk production is highly complex. One gene might control fat synthesis in the mammary gland, another regulates growth hormones, and another dictates protein quality.
So they needed the full picture.
Right. By looking at multiple candidate genes simultaneously, the researchers build a comprehensive view of the entire genetic architecture underlying the animal's dairy performance.
Okay, so let's actually look at the results. When they unleashed those molecular scissors on the DNA, what did they find? The study starts by detailing a gene called caspase, or is it casein?
Yes, caspase. It's heavily studied because it dictates the milk's protein composition, which directly impacts its cheese-making properties. When they analyzed the Ladakhi cattle, the researchers found that the A allele was the most predominant, appearing with a frequency of 0.57. However, they also found a significant presence of the B allele at a frequency of 0.36.
And based on the research, that B allele is the one specifically associated with better cheese yield and better coagulation properties, right?
Yes, exactly.
So the fact that over a third of the genetic makeup favors high-quality cheese production is a huge asset for a community that totally relies on preserving milk for those harsh winters.
It's a crucial survival trait.
But there was a curveball in this specific gene. The researchers also found a third variant, an E variant, at a small frequency of 0.06.
Yeah, the E variant is indeed the mystery in the data. The E variant actually decreases casein content, which lowers the cheese yield.
So it's worse for cheese.
Right. But more importantly, the presence of the E allele is highly unusual for native Indian cattle. It serves as a genetic clue.
A clue to what?
It strongly suggests there might have been a slight genetic admixture. In other words, some historical interbreeding likely occurred with exotic taurine cattle, particularly in the areas closer to Les City where those exotic breeds might have been introduced.
I'm going to have to stop you there. First, what exactly do you mean by exotic taurine cattle? And second, how on earth does a non-native cow end up interbreeding with cattle at 5,000 meters in the Himalayas?
It requires a bit of taxonomic context. Cattle generally fall into two main groups. You have Bossus, Taurus, or taurine cattle, which are humpless and typically originate from Europe or Northern Asia.
Think of your classic Holsteins or Jerseys.
Exactly. Then you have Bossus indicus, or Zebu cattle, which have the prominent hump on their back and are adapted to the hotter climates of India and Africa.
And the Ladakhi cattle belong to the Zebu lineage?
Yes, they do. As for how a taurine cow gets into the Himalayas, well, it's usually due to human intervention.
People moving them around?
Right. In an attempt to boost local milk production, government or agricultural programs sometimes transport high-yielding exotic breeds, like Jerseys, into urban centers like Leh City.
Oh, I see.
So what this E variant tells us is that the local Ladakhi cattle near the city occasionally crossbred with those introduced Jerseys, leaving a permanent genetic fingerprint in the population.
A microscopic Jersey intruder hiding in the DNA. That is fascinating.
It really is.
Okay, let's move to what is arguably the most relatable finding in the whole study. Something anyone listening has probably seen heavily marketed at their local grocery store. I'm talking about the beta-casein gene, or BOCCN.
Oh, yes.
This gene is the entire basis for the massive A1 versus A2 milk debate.
This is a critical point of analysis for the study. When the researchers tested the beta-casein gene, they found that Ladakhi cattle overwhelmingly possess the A2 allele, with an astonishing frequency of 0.92.
I see A2 milk selling for a premium price everywhere now. But the actual biological mechanism behind why A2 milk is considered healthier is incredibly specific. It all comes down to a single amino acid difference in the protein chain, right at position 67, doesn't it?
That is the exact mechanism. Let's look at A1 milk first, which is heavily produced by many Western commercial dairy breeds.
Okay.
In the genetic instructions for A1 milk, the amino acid at position 67 is histidine.
Histidine, got it.
The problem arises when humans drink it. As the human gastrointestinal system digests A1 milk, digestive enzymes target that specific histidine link, causing the protein to split.
And what happens when it splits?
When it splits, it releases a 7-amino acid bioactive opioid peptide called beta-casomorphin 7, which is usually abbreviated as BCM7.
Wait, so drinking a standard glass of A1 milk literally releases an opioid peptide into the human digestive tract?
Yes, and the scientific literature surrounding BCM7 is quite concerning. It is known to affect opioid receptors across the nervous, endocrine, and immune systems.
That does not sound good.
It isn't. More specifically, research has identified BCM7 as an oxidant of low-density lipoproteins, your LDL cholesterol.
Which is the bad cholesterol.
Right. The oxidation of LDL is the primary driver in the formation of arterial plaque, which leads to heart disease. So there's a substantial body of evidence linking the consumption of A1 milk and the subsequent release of BCM7 to adverse physiological responses, digestive discomfort, and broader immune system hazards.
But the milk from the Ladakhi cattle avoids this entirely. Because their DNA dictates that they are overwhelmingly A2, the amino acid at that exact same position, 67, isn't histidine, it's proline.
Yes, probably.
And based on the chemistry, proline acts like a biological block. It physically prevents the human digestive enzymes from splitting the protein at that site.
Exactly, because the protein never splits. That hazardous BCM7 peptide is never formed in the gut.
That is amazing.
That is the crux of why A2 milk is so valuable. Instead of producing BCM7, the digestion of A2 milk produces a completely different peptide that does not carry those negative cardiovascular or gastrointestinal health associations.
What?
The fact that the Ladakhi cattle carry this A2 trait at a 92% frequency makes their milk a highly desirable, exceptionally healthy nutritional source, produced completely naturally in one of the hardest environments on Earth.
Natural medicine disguised as dairy. I love that.
Mm-hmm.
Beyond the milk quality, though, the DNA also confirms some deep historical roots and specific survival traits.
It did.
The researchers analyzed the bovine growth hormone gene and found a high frequency of something called the MSPI- allele.
Yes.
They also looked at the DGOT1 gene and found the KK genotype at a frequency of 0.59. I have absolutely no idea what those random letters mean for the actual cow, though.
They are vital pieces of the puzzle, actually. Let's take the MSPI- allele in the growth hormone gene first.
Okay.
That specific genetic spelling is a classic defining marker of Indian zebu cattle heritage.
Oh, so it proves their lineage.
Finding it in high frequency confirms their ancient evolutionary lineage and proves they are deeply rooted in the native cattle populations of the subcontinent.
And what about the other one?
As for the DGAT1 gene, the KK genotype is widely recognized by geneticists as a strong indicator of a high milk fat percentage.
Which makes perfect sense when you consider the environment. If you're a calf, or a human for that matter, trying to survive winter in a freezing mountain desert, you don't necessarily need 30 liters of watery milk.
No, you'd freeze.
Right. You need energy-dense, high-fat calories just to keep your body temperature up.
Exactly. The genetic architecture is perfectly tuned to the environmental demands. But all of these findings, the zebu heritage, the rich fat content, the A2 milk, they all build up to what is arguably the most significant breakthrough of the entire paper.
Oh, lay it on me.
The researchers utilized their molecular scissors on a gene called butyrophilin, or BTN3.
BTN3.
This gene is heavily involved in the actual secretion of milk. And when they looked at the resulting DNA fragments, they discovered something entirely unexpected. They found two completely novel genotypes, AC and AE.
Wait, when the paper says novel, do they mean novel just to this region or completely new to science?
They mean completely new to science.
Wow.
These specific genetic variants, AC and AE, have never been observed in any other Indian native cattle population. Furthermore, they have never been documented in any exotic taurine cattle breeds globally.
That is incredible.
They appear to be utterly unique to the Ladakhi cattle.
So, out of all the millions of cattle across the globe, the specific spellings in the instruction manual only exist in this one isolated population, breathing thin air up in the Himalayas.
Yep.
But what does the BTN3 gene actually do? Like, how does this new spelling help them?
Well, BTN3 is crucial for the function of the mammary gland, specifically in how lipid droplets, the fats, are secreted into the milk.
Right, which we know is super high fat.
Exactly. Producing and secreting milk is an incredibly energy-intensive process for a mammal's body. Doing so in an environment where oxygen is severely limited and temperatures are freezing puts immense metabolic stress on the mother.
I can't even imagine.
While we do need more functional studies to be certain, the presence of these entirely unique
genotypes suggests that the Ladakhi cattle have evolved a highly specialized, optimized cellular machinery.
To handle that exact stress.
Right. This novel genetic scaling likely allows their mammary glands to efficiently process and secrete energy-dense milk under extreme physiological stress without compromising the mother's own survival. It's undeniable molecular proof of their unique evolution.
It's as if thousands of years of facing freezing temperatures and scarce food have literally forced their genome to rewrite its own operating instructions to survive where other animals would fail.
That's essentially what happened.
Which makes the conclusion of this study feel incredibly urgent to me, because this unique genetic purity, this masterpiece of natural engineering, is fragile.
It is highly vulnerable, and the researchers use their conclusion to issue a critical warning. In agricultural policy, there is always immense economic pressure to increase yield.
Right. More milk equals more money. Exactly. If local farmers are incentivized by well-meaning but frankly short-sighted programs to extensively crossbreed these native Ladakhi cattle with exotic high-yielding taurine breeds like Jerseys.
Just to squeeze out a few extra liters of milk.
Yeah, the consequences could be catastrophic for the breed.
Because you dilute the manual. You start mixing in pages of instructions from a cow that requires a massive caloric intake and sea level oxygen, and you just overwrite the very traits that allow the animal to survive at 5,000 meters.
It is the ultimate risk. If you crossbreed them extensively, the genetic dilution happens rapidly. Those incredible, naturally evolved survival traits, those completely novel BTN3 genotypes that help them process energy, and the overwhelming health benefits of the A2 milk, they could all be washed out of the population.
Lost forever in just a few generations.
And once that unique genetic resource is gone, we cannot engineer it back in a lab. Conserving the pure Ladakhi cattle isn't about romanticizing traditional farming. It is a highly pragmatic, necessary step to protect a biological safety net for a rapidly changing world.
We started this deep dive shivering in the freezing, oxygen-starved deserts of Ladakh, and we ended up navigating the microscopic instruction manuals of DNA.
Quite the journey.
It really is. It is remarkable how much a single, isolated breed of cattle can teach us about resilience, the mechanics of evolution, and the hidden value of what nature has engineered over millennia.
It certainly redefines how we view livestock.
Yeah.
It highlights why we must prioritize the scientific study and conservation of indigenous breeds globally before we inadvertently erase their unique genetic codes in the blind pursuit of short-term productivity.
It really makes you reconsider the value of isolation. So I want to leave you with a final thought to mull over today. If this one, remote breed of cattle up in the Himalayas, holds entirely novel genetic variations for surviving extreme climates and naturally produces some of the most uniquely nutritious milk on Earth, what other genetic gold mines are hiding in plain